Let's solve the trickiest paper 1 questions of 2018. And don't forget, before the start of the exams, we have a unique service which you could ask unlimited number of questions about the AS Biology syllabus for only $12 per month. So let's start. Which eyepiece and objective lens combination of a light microscope allows the greatest number of cells in a field of view to be seen? Now, here, greatest number of cells in a field of view, this means we have to zoom out. So the answer with the lowest magnification would be correct. The reason for this is when we zoom out, more number of cells in the field would be seen compared with if we zoom in. Therefore, the lowest magnification we have here is going to be 5 times by 10, and the answer is going to be A. And by the way, to calculate the total magnification of a light microscope, we multiply the magnification of the eyepiece lens by the objective lens, giving us 15 magnification, which is the lowest one of all the options. Question number 7. Which concentrations could be produced by serial dilution of an 8% glucose solution? So usually the method we do serial dilutions with is we use a very simple formula of M1 V1 equals M2 V2. M stands for concentration and V stands for volume. However, here we only have percentages. Therefore, we cannot really use this formula and we have to do a serial dilution by a different way than usual. Now, a trick to how to do serial dilution using percentages is the following. Now, we know that 8% is the same as 8 over 100. Now, we know that in serial dilutions, we decrease by the same dilution factor each time. So our dilution factor is 8%. Therefore, we decrease by 8% every single time. And a very easy way to do this would be to do 8 over 200. We decrease by 8% dilution factor. Then 8 over 400. Then 8 over 800, and 8 over 1600, and so on. Therefore, this would give us 8% to start with, then 4%, then 2%, then 1%, then 0.5%, and so on. Therefore, the only correct answer in this situation would be A. Question number 20. How many copies of each different DNA molecule will be found in a cell at the start of each of these cell stages of the mitotic cell cycle? So the key here is at the start. Now, we must clarify a point, is that the S phase is where the DNA duplicates. So everything that comes after S phase has duplicated DNA. Now let's start with G2. G2 is right after S phase. Therefore, it would have 2 copies of the DNA because it has been duplicated. Therefore, 2 is going to be correct. Now, during prophase, and prophase is the start of mitosis. Then metaphase, then anaphase, then telophase. During prophase, the number of DNA is still the same. It hasn't been halved or divided. From G2 to mitosis, it's still the same number. Therefore, it's also going to be 2. Now here, for cytokinesis, the key here is because the question is saying at the start of each of these stages. At the start of cytokinesis, the cell hasn't yet divided. Therefore, it would look something like this, with 2 nucleuses. Therefore, it would still contain, the full cell would still contain 2 copies of DNA before it has been divided. Therefore, the answer must be D. Question number 23. A culture of bacteria was allowed to reproduce using nucleotides containing the heavy isotope of nitrogen-15. In which positions would the DNA be found after the cells had divided once and after the cell had divided twice? Now, ideally, you should be able to know and recall the exact percentages during the exam to save time. However, I will explain the root behind the values and percentages. Now, we start with nitrogen-15, nitrogen-15. Now, in the first division, where nitrogen-14 is added, what happens is that it now turns hybrid. So it's N15 by adding nitrogen-14. It's now N15, N14, and N15, N14. Therefore, now we have a hybrid DNA with two strands being 1, 14 and 1 is 15. Therefore, this test tube actually represents the weights of it, by N15 having the greatest weight, so being at M, and N14 having the lightest weight, being at K. Now here we're somewhere in the middle because the strand contains both 15 and 14. Therefore, all of these strands would most likely be at L because this is the area at the middle. So all of it would be at L because all of it are hybrids. Now, after dividing twice, let's divide again. Therefore, we keep adding nitrogen 14. Now we have N15, N14, 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 N15, N14, and N14, N14. Now, in this case, we have two hybrids and two light chains. Therefore, in this case, as we mentioned, because these are two hybrids, there will be somewhere in the middle, which is L, and because we have now two light, N13 and N14, which are these ones, it's going to be at K because K has the lightest nitrogen isotopes. Therefore, the answer must be half at K and half at L. Because it's 2 and 2, therefore the answer must be C. Question number 37. Bacteria that infect skin wounds can become resistant to the antibiotic commonly used to kill them. Which statements explain how these bacteria could become resistant to the antibiotic? Now, how bacteria becomes resistant? During its natural life cycle, what happens is bacteria over time accumulate mutations, and then it keeps mutating all the time. Therefore, a sudden mutation could happen which could enable it to have resistance for a specific antibiotic. However, the mutation occurs first before the introducing of the antibiotic. Now let's see the suggestions that we have here to elaborate further. A. Exposure to the antibiotic causes a change in the bacteria's cell wall, preventing the antibiotic from working. Now, a huge error here is it's assuming the exposure of the antibiotic is what caused the mutation. However, the mutation to a specific antibiotic is already present without the exposure of the antibiotic, because antibiotic is not a mutagen. For example, only a single gene acquired by the bacteria could give it resistance to an antibiotic. Therefore, this is totally incorrect. Now for B, other bacteria that commonly protect the skin infections are killed by the antibiotic, allowing the infectious bacteria to survive. So, that they are killed in the first place, this does not mean that they are resistant. Therefore, this is incorrect. For C, random mutations in DNA, which is correct. Mutations happen randomly and they have nothing to do with the introduction of antibiotics, allows some of the bacteria to survive in the presence of the antibiotic. Now, let's say a few bacteria has been mutating, 5 bacteria for example, and one of these bacteria suddenly acquires an allele which actually specifically gives it resistance to that specific antibiotic. Therefore, once the antibiotics introduced, the other 4 are killed and one of them is able to survive and reproduce and pass on this allele. Therefore, C is totally the correct answer. Now for D, the antibiotic causes the bacteria DNA to mutate. Now, another error is here assuming that the antibiotic is what causes the mutation. Therefore, this is incorrect and the answer is going to be C. Question number 40. A vaccine is used to create artificial active immunity. After being given a vaccine, it would take a period of time before a person develops long-term immunity against the disease. Which statement about this period of time explains the delay? Now, first of all, we have to explain what vaccines do in the first place. Vaccines introduce antigens. Now, once the antigens are inside the body, there are B cells with specific B cell receptors. Therefore, a specific B cell, when encounters this specific antigen, they bind, they form a complex. Therefore, that B cell is actually stimulated to now divide and produce plasma cells specific to that antigen and memory cells specific to that antigen, and this is the primary immune response, where the antigen has never been introduced to the body before. Now, the formation of plasma and memory cells actually takes time. This process takes time. However, in the secondary immune response, the memory cells already produced by the primary immune response before are able to divide quicker to form plasma cells and produce antibodies. So the plasma cells is what produces antibodies at a faster rate. Now let's see the suggestions that we have here. A, no memory cells have been produced from B lymphocytes. Yes, because memory cells are not yet produced. Now this process, as I said, it actually takes time. Therefore, this is what causes the delay. Therefore, the A is going to be the correct answer. Now for B, no plasma cells have been produced from the B lymphocytes. Now, even if this is a unique antigen, still plasma cells are going to be produced eventually. Therefore, this is actually incorrect to assume that no plasma cells are being produced. Now for C, the primary immune response has not produced enough antibodies. There's no such thing as not produced enough antibodies. This does not cause the period of time delay that has been mentioned. Therefore, this is incorrect. For D, the secondary immune response has not produced enough antibodies. Now, this is totally incorrect because you just said that during the secondary immune response, antibodies are actually produced at an even greater rate than the primary immune response. Therefore, the only logical answer in this case would be A, that memory cells take time to form in the primary immune response. This is why a delay has been taken place.